Hello. The second lecture on renal physiology by Dr. Fahim. In the last lecture, we talked about the ability of the kidney, how to clear the waste product from the blood, and how to maintain the body essential water, and how do electrolyte balance. Is dependent on renal blood flow and tubular absorption. In this lecture, we will talk about glomerular filtration and tubular secretion, and is controlled in the nephron by renal function. Tubular reabsorption. The body can't lose 120 ml of water containing essential substance every minute. When the plasma ultrafiltrate enters the proximal convoluted tubules in the nephrons, the cellular transport mechanisms start reabsorbing the essential substance and water. Reabsorption mechanisms. The cellular mechanisms and tubular reabsorption are active and passive transport. Firstly, the active transport occurs by combination between substance and carrier protein in the renal tubular cell membrane. And this interaction transfers the substance across the cell membrane and back into the bloodstream by electrochemical energy created. Active transport is responsible for reabsorption of glucose, amino acid, and salts in proximal convoluted tubule, chloride in ascending loop Henle, and the sodium in proximal and distal convoluted tube. Secondly, the passive transport. The passive transport is the movement of molecules across the membrane due to the difference in their concentration or the electrical potential on the opposite side of the membrane. Passive reabsorption of water takes place in all the parts of the nephrons except the ascending Henle loop, where the walls are impermeable of water. Passive reabsorption of urea takes place in the proximal convoluted tubule and the ascending Henle loop. Passive reabsorption of sodium meets the active transport of the chloride and the ascending Henle loop. Active transport and the passive transport are influenced by the concentration of substance being transported. When the concentration of substance in the plasma is abnormally high level, the filtrate concentration exceeds the threshold maximal reabsorptive capacity (TM) of the tubule, so the substance begins appearing in the urine. The plasma concentration where the active transport stops is known as the renal threshold. The renal threshold is 160 to 180 mg per deciliter for the glucose. So, it appears in the urine when the plasma concentration reaches this level. Knowledge of the renal threshold and the plasma concentration can be used to distinguish between excess fluid filtration and renal tubular damage. For example, glucose appears in the urine of the person with normal blood glucose level is the result for tubular damage and not for diabetes mellitus. Active transport of the filtrated sodium out of the proximal convoluted tubule is combined by passive reabsorption of an equal amount of water, and the fluid leaving proximal convoluted tubule still maintains the same ultrafiltrate concentration. Tubular concentration. The concentration process starts in the descending and the ascending Henle loop, where the renal filtrate is exposed to the high osmotic pressure gradient of the renal medulla, where water is reabsorbed by osmosis, and in the descending Henle loop, sodium and chloride are absorbed in ascending loop. When the ultrafiltrate plasma is high concentration, the reabsorption of water is prevented by water impermeable walls of the ascending loop. And this selective reabsorption process is called countercurrent mechanism. Countercurrent mechanism work on maintain the osmotic gradient of the medulla. The sodium and chloride reabsorption in the ascending loop prevents the dilution of the interstitial medullary by water reabsorption from the descending loop. Maintenance of the osmotic pressure gradient is essential for the final concentration of the filtrate when it reaches to the collecting duct. The concentration of filtrate from the ascending loop is low concentration due to the reabsorption of salt, not the water, in this part of tubule. Reabsorption of sodium continues under the control of hormone aldosterone, which regulate the reabsorption according to the body need of sodium. Collecting duct concentration. The final concentration of the filtrate begins with the reabsorption of water in the distal convoluted tubule and the collecting duct. When the diluted filtrate contact with the higher osmotic concentration lead to passive reabsorption of water. Reabsorption process depend on the osmotic pressure gradient in the medulla and antidiuretic hormone, vasopressin hormone, ADH. ADH influencing the high level of ADH increase permeability, resulting in increased reabsorption of water and low volume concentrated urine. Absence of ADH increase the wall impermeable of water, causing a large volume of diluted urine. Production of aldosterone is controlled by the body's sodium concentration. Production of ADH is determined by state of body hydration. Chemical balance in the body determine of urine volume and urine concentration. As we see, when the body hydration is high, ADH is low and the urine volume is high. In the opposite side, when the body hydration is low, the ADH is high and urine volume is low. Tubular secretion. Tubular secretion makes two major functions. Elimination of waste product non-filtrated by glomerulus. Regulation of acid-base balance in the body through the secretion of hydrogen ion. Medication and other foreign substances can be filtrated by glomerulus because they are bounded to plasma protein. But when these protein-bounded substances enter the peritubular capillaries, they make strong affinity for tubular cell and dissociate from their carrier protein, were transported into the filtrate by tubular cell. The major site for removal of these non-filtrated substances is proximal convoluted tubule. In this slide, we see three diagrammatic. The first one is how to reabsorption of filtrate bicarbonate. And the second, how to secrete hydrogen ion combined with phosphate. And the third, secretion of hydrogen ion combined with ammonia produced by tubules. Acid-base balance. Maintaining the normal blood pH 7.4 as by eliminate the excess acid formed by dietary intake or the body metabolism. The buffering capacity of the blood depends on the bicarb ion, which are filtrated by glomerulus and fast returned to the blood to maintain pH. The secretion of hydrogen ion by the renal tubular cell into the filtrate prevents the filtrated bicarb from being excreted in the urine and returned back to the plasma. This process provides almost 100% reabsorption of filtrated bicarb ion and occur in the proximal convoluted tubule. Hydrogen ion is a small molecular size, so it's easy to filtrated, reabsorbed, and excretion depend on tubular secretion. The secreted hydrogen ion combine with filtrated phosphate ion instead of bicarb ion and excreted rather than reabsorbed. The additional excretion of hydrogen ion is accomplished through the reaction with ammonia produced and secreted by the cell of this convoluted tubule. In proximal convoluted tubule, the ammonia is produced from breakdown of glutamine amino acid, and the ammonia react with hydrogen ion to form ammonium ion, then excreted in the urine. All these three processes occur simultaneously at a rate determined by acid-base balance in the body. Disorders in this secretory function can lead to metabolic acidosis, renal tubular acidosis, or inability to produce acidic urine. With my best wish for success, Dr. Fahim, thank you. This is the second lecture for us.